Haploid and diploid cells are two types of cells in the cell process. Diploid cells contain two complete sets of chromosomes, while a haploid cell splits off of the diploid cell and contains half of the number of chromosomes that the diploid cell does. Diploid cells reproduce through mitosis, whereas haploid cells reproduce through meiosis. Diploid cells can be found in cells such as your skin, blood, and muscles, whereas meiosis cells are used in sexual reproduction, such as like the ova and the sperm. This is a cell cycle. Although a lot is said and talked about mitosis, it actually spends over 90% of its time in the interphase, which is the G1, the S, and the G2, where the cell grows, and then more specifically in the S phase, where the chromosomes are duplicated and it also grows. As previously mentioned, the cell spends most of its time in interphase. However, the most important and active part of the cell cycle is the mitotic phase. First, we're going to have prophase. This is when the chromatid fibers become more tightly bound. The nuclei disappear, sister chromatids begin to appear, and the mitotic spindle begins to form, and the centrosomes move away from each other, all located right here. The next cycle is going to be called prometaphase. Here, the nuclear envelope fragments, as you can no longer see it. Chromosomes have become more condensed. Now, kinetochores can be found at the center of the chromosomes, and some of the microtubules attach to the kinetochores, as demonstrated. After this phase is metaphase. Metaphase is the longest stage of mitosis. Now, chromosomes are in centromeres at the opposite sides of the cell. Chromosomes convene on the metaphase plate, and the kinetochores of the sister chromatids are now fully attached to opposite poles. After the cycle called metaphase comes the cell cycle called anaphase. Anaphase is the shortest stage of mitosis. In this phase, the two liberated daughter chromosomes begin to move towards opposite sides of the cell, as demonstrated. The cell elongates as the non-kinetochore microtubules elongate, and then at the end of anaphase, the two ends of the cell have equal numbers of chromosomes. After anaphase comes telophase. Telophase is when the two daughter nuclei form in the cell. The nuclear envelope begins to appear, and the nuclei reappear. These chromosomes then become less condensed, and in this stage of mitosis, the division of one nucleus into two is now complete. After this comes cytokinesis. Cytokinesis is the division of the cytoplasm, and this is when the two daughter cells appear. In animal cells, the formation of the cleavage furrow occurs.
As Ellie talked about earlier, these are the stages of mitosis. The first is interphase, which is where it is a diploid cell and there's 3 chromosomes. Interphase is 90% of the times that a cell is spent. In the second phase is prophase. This is when the DNA replicates in pairs. And then the next is metaphase. And this is when the nucleus will dissolve. There's no nucleus as shown in this one. And the spindles will attach to the chromosomes. The next is anaphase. And this is when the spindles begin to pull the sister chromatid to the opposite poles. And telophase is when the spindles disappear and the cell division will begin. And then last is cytokinesis, and this is when the cell divides and 2 daughter cells are now formed. In this image shows the spindle fibers in a cell. These are the spindle fibers at the end. These are important protein structures that divide genetic material in a cell. The spindles are also important because they divide chromosomes into daughter cells in both mitosis and meiosis. The mitotic spindle in mitosis, and then the meiotic spindle in meiosis, and it pulls sister chromatid to opposite poles so the cell can begin to divide.
Cytokinesis is the division of a cell at the end of mitosis or meiosis, bringing about the separation into two daughter cells. So in this slide, we'll talk about animal and plant cells and cytokinesis. So in an animal cell, cytokinesis occurs through cleavage and a cleavage furrow, and the cell plate, unlike the plant cell, is not formed and the wall formation is absent. So the cleavage furrow happens and then two daughter cells are formed. In the plant cell, it involves a cell plate and the vesicles derived from the Golgi apparatus move along microtubules to the middle of the cell where they coalesce, producing a cell plate, which is right here. The cell plate enlarges until its surrounding membrane fuses with the plasma membrane along the perimeter of the cell, and then in the end, two daughter cells are produced. In this next point, I'm going to talk about somatic cells versus gametes. A somatic cell contains the diploid, which is two complete sets of chromosomes, one from each parent, such as this, while the gamete contains the haploid, or has a single set, just like this. It's unpaired. Somatic cells are produced through mitosis, while the gametes are produced through meiosis. And an example of the somatic cells would be like your muscle cell, a nerve cell, or a stomach cell. And then gametes would be anything such as a sperm cell or an egg cell. These are the vehicles that transit genes from one generation to the next. So they're just kind of like transportation. While a somatic cell are any cell of a living organism other than the reproductive cells.
In this next slide, we're going to talk about meiosis 1, which starts off an interphase just like mitosis, which is where the cells grow and begin to form. And after that, the next phase is prophase. Prophase is where the chromosomes begin to condense and homologs loosely pair along their lengths and are aligned by a gene. And it also is the crossing over, which is the exchange of responding segments of DNA molecules by non-sister chromatids. And this is completed while homologs are in synapsis, held tightly together by proteins along their lengths. Synapsis ends in mid-prophase, and the chromosomes in each pair move apart slightly, as shown above in this picture. The next phase we have is metaphase 1, which is where pairs of homologous chromosomes are now arranged on the metaphase plate with one chromosome in each pair facing each pole. Both chromatids of one homolog are attached to the kinetochore microtubules from one pole. Those of the other homolog are attached to the microtubules from the opposite pole. And then after metaphase 1 is anaphase 1. This is the breakdown of proteins responsible for sister chromatid cohesion along chromatid arms and allows homologs to separate. The homologs move toward opposite poles and are guided by the spindle apparatuses. So there's the poling in the picture. The sister chromatid cohesion persists at the centromere, causing chromatids to move as a unit towards the same pole. Next is telophase 1, and cytokinesis. At the beginning of telophase 1, each half of the cell has a complete haploid set of replicated chromosomes. Each chromosome is composed of two sister chromatids. One of both chromatids include regions of non-sister chromatid. So in this picture, you can see the non-sister chromatids connected. Cytokinesis, which is the division of the cytoplasm, where you can see where it starts to divide in telophase, and then in cytokinesis, it's completely divided. In animal cells, a cleavage furrow is formed. In plant cells, a cell plate forms. In some species, chromosomes decondense and the nuclear envelope reforms. No replication occurs between meiosis 1 and meiosis 2.
And now we are going to talk about meiosis 2. It starts with prophase number 2, and this is when a spindle apparatus forms. So in late prophase 2, chromosomes move toward the metaphase 2 plate, which is right here. So metaphase 2, the chromosomes are positioned on the metaphase plate as in mitosis. And because of the crossing over in meiosis 1, the two sister chromatids of each chromosome are not genetically identical. So next we'll move to anaphase, number 2, and this is the breakdown of proteins holding the sister chromatids together at the centromeres, allowing the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. So right here, they're starting to split and like the two cells are starting to form. In telophase 2 is when the nuclei forms, the chromosomes begin decondescending, and cytokinesis occurs. So the meiotic division of one parent cell produces 4 daughter cells, such as this, each with a haploid set of unreplicated chromosomes. So each of the 4 daughter cells is genetically distinct from the other daughter cells and from the parent cell. Like I said earlier, because they do not have, because they're not genetically identical.
Homologous chromosomes versus sister chromatids. These two definitions and integral parts of the cell cycle are commonly confused with each other. So, homologous chromosomes, as demonstrated over here, are 2 chromosomes, one of the paternal origin and one of the maternal origin, that are identical in appearance, whereas the sister chromatids are 2 identical copies of a single replicated chromosome that are connected by the centromere. Relating back to the homologous chromosomes and the sister chromatids, the synapse and tetrads are an important part of the cell cycle. The synapse, as demonstrated here, is the pairing of 2 homologous chromosomes that occurs during meiosis. It takes place during prophase 1 of meiosis, to be more specific. Tetrads is a 4-part structure that forms during the prophase of meiosis and consists of 2 homologous chromosomes. Tetrad, synapse.
Thanks for learning Chapter 12 of AP Biology with us.